Hey folks, this is Eric. I'm just going to give a moment to see if anyone else is slowly trickling in. We are recording this, so in a bit we'll just get started, and if folks aren't able to make, we'll have this recording available for public sharing afterwards. Okay, I'm just going to go ahead and get started because I don't see any other notifications on the side. So hello everyone, my name is Eric Sid. I am one of the program officers within our Rare Diseases Research Innovation at division. Sorry, I forgot to start with that. At NCATS, and I have the pleasure of talking to you today about the second phase of our OligoTox challenge. So I'm going to go ahead and walk us through some slides. Most of this information is already found on the challenge proposal itself, which you can find both on nih.gov as well as a link on NCATS's website as well. So that would all come at the end of this. To get started, I'm just making sure you're seeing my screen progress. Yes. So our agenda today, we're going to be going over the challenge goals and background. We've already presented previously for phase 1, so much of this will be basically riding on the tail of that. We're going to go over more of phase 2 for this webinar. We're also going to go over the schedule, the prizes, and the submission requirements for phase 2, briefly on the evaluation judging, as well as we wanted to really keep most of the time available for any questions that you might have. So again, those of you guys that have previously attended our previous webinar, this is some of this will be a repeat. As a background for this challenge, oligos or oligonucleotides are therapeutics that are of an emerging modality that has been seeing an increase in number in drug development as well as an approval with rare genetic diseases over the last several years. These are not new types of modalities. Oligonucleotides have been around for quite a while. Both the FDA as well as the NIH are interested in specifically now around NAMs or new approach methodologies that can reduce the use of animal testing in activities such as preclinical safety studies and can leverage technologies such as organoids, other types of in vivo human approaches that can replicate structure and functions of human organs. In recent years, we've really witnessed a dramatic increase in the use of advanced computational methods for in silico testing. So really, a lot has been happening in this space around addressing complex biological problems. Within that, what this challenge is focusing on, NCATS is trying to incentivize the generation of high quality and publicly accessible open data sets that can really make use of in vitro human systems and can be used to advance predictive models for oligotoxicity. Some of the toxicities of particular interest to us with oligonucleotides include hepatotoxicity, nephrotoxicity, thrombocytopenia, complement activation, coagulopathy, immunotoxicity, chronic neurotoxicity, and hydrocephalus. And again, more information about this can be referenced in our challenge proposal documents. Now, when we previously had our first webinar, it was for the phase 1, which ended, and we actually had 8 winners announced. You can see a list of winners on the websites that I'll list at the end of this. The first phase was focused on ideation. This second phase is really the meat of this challenge. This second phase will focus on data generation. It has already started, and you have until the end of this calendar year, December 31st, to submit your application. The aim of the phase 2 is to make publicly available a high quality open data set that has demonstrable use in developing and training predictive models for oligonucleotide toxicity. In this round, we're looking to award a maximum of 2 winners, full prize winners, with up to $100,000 each, and 4 runner-ups with a prize of up to $50,000 each. The requirements for phase 2 include 4 parts. And again, this information is almost straight from the challenge proposal document. So please refer to that if you have any questions and details that you wanted to look over afterwards. The 4 parts that are required for this mission include a narrative document, which may include things such as an executive summary of the data set you generate, information about the positive and negative controls that were included, summary of the main findings and conclusions of your approach, a description of how the data was produced, including descriptions of relevant experimental design, data acquisition, and any computational processing of raw data that was performed. A description of how indicators and predictor variables for oligotoxicity being measured, as well as their distribution and the distribution of predictor variables amongst tested oligos. A discussion of how the results address a gap in the publicly available data relating to one or more aspects of oligotoxicity, and a discussion of how data could be used to develop a predictive model of one or more aspects of oligotoxicity. The other parts include a methodological document that provides information on the materials and methods used to generate this data set. This should include the methods used to verify and characterize the oligo identity. A public access and dissemination plan that describes how winners will disseminate information about their solution and make their solution, as well as the knowledge necessary to use their data sets, openly available for research purposes to the public. Any terms for data access and data use should be defined in allowing for open and public access, such as a Creative Commons license or other types of licensing scheme. Lastly, the data set itself. We will want access to the raw data set. It should also be supplemented with a data dictionary, information such as a schema documenting all of the metadata, and you should include the sequences of all the oligos that you've tested with this data set, as well as the location of all chemical modifications in each of these oligos, data on the purity and characterization of each, and any additional metadata that will be helpful to evaluate. Evaluation and judging. Entries that comply with eligibility and submission requirements will be scored for their scientific value, experimental approach and design, data translatability, and then proposed dataset management. This information will be evaluated by a judging panel that will include federal employees from the NIH as well as other government agencies and some non-federal subject matter experts to ultimately determine winners that will be assigned confidence ratings for how well submitters are addressing these 4 factors. The ability to solve the challenge. Is this appropriate in advancing predictive modeling of oligotoxicity? The potential impact for this data set to address some of the information gaps in the field. Feasibility and rigor, the viability and quality of this data set itself and how it was generated, as well as transparency and reproducibility, adherence to NIH scientific data sharing policies and best practices are how we would evaluate this. Here are a couple of important dates to keep in mind. Phase 2, the submission ends itself again at the end of this calendar year, December 31st. The winners will be announced by March 15th of the next year. Lastly, I'm just going to have this last slide here as we shift over to the Q&A part. If you have additional questions or are looking for additional information that we don't answer here, I would refer you to this link, which is to the NCATS page for our different active challenges, specifically the OligoTox challenge will be there. You can also send your questions to our inbox here at NCATS OligoTox at mail.nih.gov. So with that in mind, I wanted to also bring in my 2 colleagues that are going to help me with answering questions. Our division director, PJ Brooks, as well as one of our other extramural officers, Alumit. And so we just wanted to now open it up to questions that you may have. And you can either raise your hand or submit them through the Q&A.
I guess there are none.
Okay. Yeah. Seeing none. Oh, we have one hand raised. Please go ahead. If you can unmute yourself. You need to. I think you might need to unmute yourself. Yeah. Oh, it's not letting. Try again right now. I think you might have access. Wednesday, if you want to make it so that people can unmute themselves too.
Yes, we've enabled your mic, so you should be able to unmute.
And then we have another question while we're waiting for that. PJ, are the results from phase 1 being shared?
I mean, I think we've announced the winners and titles. So in that regard, yes.
And maybe I should just clarify that unlike the submission of grants to the NIH, when it comes to challenges and prize competitions, the applicants are not receiving any reviews or summary statements or any feedback back.
Okay, we have one more. Would it help our submission to include example AI modeling notebooks, train test splits, and reproducible feature engineering code to show the dataset can support predictive modeling? PJ, again, feel free to chime in. I would say that it doesn't hurt to include more information that's focused on, again, the goal of this challenge in order to be able to evaluate whether or not your data set is of appropriate use for predictive modeling, as well as any information on how your data set was generated or how you would propose it being used. Now, that's going to really depend on the specific submission. So can't really say specifically if that would be appropriate for everybody in general. Again, PJ, I don't know if there's anything else you would want to note.
I mean, I think that this seems a bit above and beyond what we're requiring. But I think if there's, so you can put some of this in there as examples. But I think you really want to respond to the requirements as written on the prize competition opportunity.
Okay. Any other questions? We'll give one last chance for it. All right. And again, remember, you can also follow. Oh, sorry. Go ahead, Peter.
No, I say, I guess not. No more questions.
Yeah, and you can follow up with us on that email if you have any questions. So thank you all for attending. Again, this is a quick one, but hopefully all the information we have out there and answering most of your questions already. Thank you.
Thank you.
Thank you.